Hello, dear students. In last lecture, we have discussed about the morphological characteristics which are used in the classification of microorganisms. Now, in this lecture, we will discuss about the what are the cultural characteristics which are used in classifying different microorganisms or grouped into different on the basis of their cultural characteristics. So, to define the cultural characteristics, we require that particular organism uh, in pure form. So, pure cultures of organism can be studied by growing them on a wide variety of culture media. So, cultural characteristics are nothing but the growth pattern of that particular organism on different culture media. So, under appropriate cultural conditions, organisms show characteristics, type of growth, and such observations are useful in the identification of the organism. Growth characteristics are commonly observed on the following types of culture, like for growing the particular organism on particular media, the organism shows the characteristics growth pattern on that particular media, so that that particular pattern of that organism is useful or can be used for the grouping of that particular organisms or identification of that organism on the basis of the growth pattern. So what type of media we are using and what type of growth pattern that organism shows on that particular media is very useful for the cultural characteristics and also for the identification of that particular organisms. So uh, to study the cultural characteristics, we discussed the colonies of that particular organism on a solid media, or you can say on plate culture, particular growth pattern of that organism on liquid media, growth of that particular organisms on agar slants, growth of that particular organism in agar stab culture, particular organism shows the cultural characteristics or pattern on gelatin stabs. So these are different culture types, or we can say different types of culture on which that particular organism shows a characteristics growth pattern and can be used for the identification of that organism. Description of colonies on solid media should include when we inoculate that particular organism on a particular culture media, the colony characteristics we have to note down, or we have to record that particular data on that characteristics group pattern. And that description of colonies on solid media should include, or we have to note down the different characteristics like size of the colony, shape of that colony, elevation of that particular colony, surface or we can say surface appearance of that particular colony, either it is smooth surface or rough surface, we can say. Color of that colony, sometimes many organisms produce a different pigment. So apart from that, also different organisms shows quite different colors on a particular media. So we have to report that particular what type of color that colony shows and appearance by reflected and transmitted light. So with respect to opacity, we have to also discuss. Similar information can also be secured from agar slant and agar stab culture. So these characteristics we can use for cultural characteristics. These are nothing but the some representative plates that we have taken, in which different organism shows different types of growth pattern on particular media. So here we can say nothing but a sticking of particular organisms on that particular media. So, or we can say Maikonki agar. It is nothing but a nutrient agar. And it is nothing but the growth pattern of E. coli, Klebsiella species, Enterobacter species. These are nothing but the Sericea species or Sericea marcescens. These are Cetrobacter growth pattern, colony of Proteus. This is nothing but the growth pattern of Shigella species, and these are Providentia. These are nothing but the characteristic growth pattern that we have representatively taken for the to understand the colony pattern of that particular organisms. So when we inoculate that particular organism in a solid media, we have to study their colony characteristics with respect to their size, shape, elevation, color, margin. So in this table, we can see if we observe in case of we have prepared a table for with respect to margin, color, elevation, texture, and shape of that particular colony. So if we observe this type of margin, we can call it nothing but the curl type of margin. If we found this nothing but the smooth or entire type of margin, we record as a entire type of colony. This is nothing but the filamentous type of colony margin. if we observe this wavy type of margin, we can call it as an undulate or wavy type of margin of that particular colony. It is nothing but the lobate type of pattern, colony pattern of that particular organism. It is eros or serrated type of margin of that colony. When we discuss about the on the basis of color, we can distribute that particular organism into either that orange color colony or red or pink color colony, black type of colony, brown, opaque, or milky. So with respect to color, we can differentiate or report that particular color of that colony. When we observe closely that elevation of that colony, we can see these different types of elevation pattern of different organisms. So if we observe this type of raised type of appearance of that particular colony, we can report it as nothing but the raised type of elevation, or it is quite upward from the surface of that agar plate. So we report it as nothing but the raised type of elevation. It is nothing but the abundant type of elevation where at the center that colony are quite move upward on that surface of that agar. It is nothing but the flat type of elevation we can report. It is nothing but the convex type of elevation of that colony. It is nothing but the pulvinate or cushion shape elevation of that colony we can report. So these are nothing but the different elevation pattern we can observe on a particular media. When we talk about the texture of that colony, we can see a slimy or moist type of texture, matte or brittle type of texture pattern, shiny or viscous type of pattern, dry or nipple type of colony pattern, transparent type of colony. Or apart from that, if we observe another different types of pattern, we have to report that. If you observe with respect to shape, we can record it as nothing but the round shape of that particular colony. These are nothing but the small round shape, but very small colonies, we can call it as nothing but the punctiform of that shape of that colony. It is nothing but the rhizoidal shape of that colony. These are nothing but the filamentous pattern of growth pattern of that particular colony, or that filamentous and this filamentous here at the center they are blank. So that type of colony we can observe. So we are included into filamentous type of colony. These are nothing but the irregular as it shows in it type of shape. So they are irregular as counted, and these are nothing but the spindle shape of colony shape. So on that basis, we can prepare a table and we can report the colony characteristics of that organism on a solid media. Now, when we inoculate a particular organism in liquid media, we can observe another different types of growth pattern of that organism in a liquid media. So if we inoculated a broth culture from that organism, we can see different types of growth pattern of that organism. Suppose here is nothing but the uninoculated, or we can say control tube, where there is no inoculation of any organism or carried out. If we inoculated that particular organism in another broad tube and we found this type of colony, or at the surface of that broth we can found a growth of that organism, we can see it is nothing but the pellicle shape of growth pattern of that organism in a broth media. If we observe all the broth are cultural growth are settled down at the bottom, we can say it is nothing but a sediment type of pattern of that culture in a broth media. If we observe a uniform turbidity or uniform growth pattern of that organism, we can call it uniform turbidity. Or if we form flocule formation of that particular organism, so we can record it as a flocculent type of growth of pattern of organism. If we inoculated that organism on a agar plant, so we observe it is nothing but the filiform type of growth pattern. It is nothing but the echinulate type of pattern, growth pattern of agar plant. It is nothing but the beaded type of growth pattern. It is nothing but the effuse type of growth pattern of that organism on its plant. It is nothing but the arborescent type of growth pattern, and it is called as the rhizoidal type of growth pattern of that organism. So each organism shows a different or characteristic growth pattern on a particular solid, liquid, or you can say slant culture media. So with respect to that, they are quite helpful for the identification of organism. Now, apart from the characteristics growth pattern, organisms are classified on the basis of their physiological characteristics, like their requirement for the growth. So physiological tests which are commonly used in the identification of the organisms are but their requirement for the growth, like with respect to temperature, with respect to oxygen. So on the basis of growth pattern, the organism which are show a quite or required temperature between the range 0 to 20 degrees Celsius, that organisms are categorized as a sacchariophilic organism. The organisms which require 20 to 45 degrees Celsius temperature for their optimum growth, they are categorized into mesophilic type of organisms. Thermophilic organisms are able to or show optimum growth in the temperature range between 45 to 65 degrees Celsius. Apart from that, thermodynamic organisms, we show their optimum growth pattern between the range of temperature that is 65 to 100 degrees Celsius. So with respect to their growth pattern, or different organisms require different temperature for their growth. So they are categorized into saccharophile, mesophile, thermophilic, or thermophilic. Apart from their oxygen tolerance or oxygen requirement, they are again categorized into different groups. So strictly aerobic organism, or for the growth of that organism, they require oxygen. They are called as strictly aerobes, or you can say obligate aerobes. It is again another group of organism, or you can categorize that organism into that group. Strictly anaerobe, that means that organism could not survive in the presence of oxygen, or we can say oxygen could be lethal for the growth of that organism, and that organisms are called as strictly anaerobes. Facultative anaerobes, that means they doesn't require oxygen but can grow in presence of oxygen. That means in presence or absence, they can survive. They are called as facultative anaerobes. Apart from that, the fourth group, nothing but the microaerophilic. That means the range of 2 to 10% oxygen for their growth is required. So on the basis of oxygen requirement, organisms are grouped into different groups. So on the basis of that biochemical characteristics and physiological characteristics, we can group different organisms into different groups, and they are quite helpful for the classification of organisms. Next characteristics we'll discuss in next lecture.